Hello students, today we learn about genetics. Let's start. Genetics, the exploration of the working and major codes of variation and heredity is termed as genetics. The groundwork on which heredity stands is known as inheritance. It is defined as the procedure by which characteristics are handed down from one generation to the other. Gregor Johann Mendel is known as the father of modern genetics for his discoveries on the basic principles of heredity. Variation. Variation, as the name suggests, is the amount of dissimilarity that exists in between children and their parentages. It can be determined to keep in view the behavioristics, cytological, physiological, and morphological characters of individuals fitting into similar species. Major reasons that variation are: first is genetic chromosomal rearrangement, second is mutated genes due to the influence of the ecosystem, third is crossing over. Mendel's law of inheritance. Mendel conducted hybridization experiments on garden pea Pisum sativum for 7 years and proposed the law of inheritance in living organisms. Selection of pea plant. The main reason for adopting garden pea Pisum sativum for experiments by Mendel were: first, pea has many distinct contrasting characters. Second is lifespan of pea plant is short. Third is flowers show self-pollination, reproductive holes being enclosed by corolla. Fourth is it is easy to artificially cross-pollinate the pea flowers. The hybrids thus produced were fertile. Working method. Mendel's success was also due to the meticulous planning and method of work. First, he studied only one character at a time. Second, he used all available techniques to avoid cross-pollination by undesirable pollen grains. Third, he applied mathematics and statistics to analyze the results obtained by him. Mendel selected 7 contrasting characters of garden pea. First column character, second column contrasting character, dominant, recessive. First is stem height, tall, dwarf. Second, flower color, violet, white. Third is flower position, axial, terminal. Fourth is pod shape, inflated, constricted. Fifth is pod color, green, yellow. Sixth is seed shape, round, wrinkled. 7 is seed color, yellow, green. This is a table of Mendel's law of inheritance. Artificial hybridization. Mendel conducted artificial hybridization cross-pollination using true breeding pea lines. True breeding lines are those that undergo continuous self-pollination and shows stable trait inheritance. Hybridization experiments include emasculation, remove of another, and transfer of pollination. Mendel's experiment procedure. First, he observed one trait or character at a time. For example, he crossed tall and dwarf pea plant to study the inheritance of one gene. Second is Mendel hybridized plants with alternate forms of a single trait, monohybrid cross. The seeds produced by these crosses were grown to develop into plant of filial 1 progeny or F1 generation. Third is he then self-pollinated the tall F1 plant to produce plants of filial 2 progeny or F2 generation. Fourth is in later experiments, Mendel also crossed pea plant with two contrasting characters known as dihybrid cross. Fifth is Mendel self-pollinated the F2 plant also. Inheritance of one gene, monohybrid cross. Mendel crossed tall and dwarf pea plant and collected all the seed obtained from this cross. He grew all the seed to generate plant of first hybrid generation called F1 generation. He observed that all the plants are tall. Similar observation was also found in other pair of traits. Mendel self-pollinated the F1 plant and found that in F2 generation some plants are also dwarf. The proportion of dwarf plants is 1/4 and tall plants of 3/4. Genes. Mendel called the factors that passes through gametes from one generation to next generation. Nowadays it is called as genes, unit of inheritance. Genes that code for a pair of contrasting traits are known as alleles. Monohybrid ratio. Mendel also proposed that in true breeding tall and dwarf variety, allelic pair of genes for height is homozygous TT and TT. Capital TT and capital T and small t or small tt are called genotype and tall and dwarf are called phenotype. The hybrid which contain alleles which express contrasting traits are called heterozygous. Test cross. Test cross is the cross between an individual and dominant trait and a recessive organisms in order to know whether the dominant trait is homozygous or heterozygous. Dominant phenotype unknown genotype. Recessive phenotype known genotype. Predictions sperm XX. This is a genetic part 1 video. Next video we will upload soon. Thank you for watching this video and please subscribe the channel and press bell icon for latest video. Thank you so much.